All right, so today, first off, I already asked, but no one asked any questions about the pending quiz that you have. That's chapters 7, 11, and 12. And so if no one has any questions, then I'm going to start talking about chapter 13 and about the 2 lab modules. So, okay, so can you unmute and ask your question?
Yes.
Okay.
Can you hear me?
Yep.
Okay. I just wanted to know, will we need to know what microorganisms are like the least resistant and most resistant to antiseptic
Yes.
technique? So we need to know them in order, or just like
both? You need to know them in order that was pictured in the,
yeah.
You need to know them in order, and then you
Okay.
need to know for each disinfectant and for each chemical, for each physical method, for each mechanical method, which organisms that method works for.
Okay. That's it.
Yep. Yep. That's the short answer. Yes.
Okay.
All right. Does anyone else have a question about the exam before I start talking about the lab modules and the chapter 13? There's a couple things that you need to know kind of almost ahead of time for chapter 13. Okay, so I'm just gonna go then. So chapter 13 begins sort of a new section for us, and it'll be the next lecture quiz will be chapters 13, 14, and 15 together. And they are all on this section of what we would call microbe-human interactions is chapter 13, and then chapter 14 is what we would call innate defenses. So chapter 13 is all about the things that the microorganisms have that benefit them and allow them to cause an infection, which obviously is going to impact the host in a negative way. And then chapters 14 and 15 are particularly human host, the human host response. So the first chapter we would call innate response or the non-specific response, that's chapter 14. And then 15 is about the immune system. So if you've covered the immune system in anatomy and physiology already, AP2, great, that'll be easy for you. It'll be the second time around. If you've not done the immune system before, it is complicated. I'll give you as much support as I can, but it's going to take a lot of reading and a lot of reviewing. For chapter 13 in particular, I usually ask a large section of the quiz for 13, 14, and 15, not the one you're taking tomorrow, but the next one coming up. I usually ask a large section of that as matching terms, terminology. So what I would suggest is particularly for chapter 13, I would go through the chapter and I would look for the words in bold, because those are the words that I'm going to choose as the vocabulary words that I'm going to ask you on the quiz. There are some that I would toss out, and I can make a list for you and post it. But most of the words, I believe there's 90 some of them, a lot of them are variations of the same word, and some of them are words that you already know from just being alive, working in perhaps a medical capacity of some sort, or just they're pretty common sense sorts of words. And what I mean by that is, for example, your book lists all in bold, toxin, toxosis, toxogenicity, and all of those words basically mean the same thing, right? They're all about, I would put those all on one card. Those are all about how a particular substance that a bacteria or virus, a bacteria produces, how they are toxic or poisonous to the host. So same with like pathogen, pathogenicity, those words, right? One's a process, one's the actual noun. So I would put those words on the same flashcard, but I would make flashcards for the vocabulary words for chapter 12, the words in bold, not the words in blue that are the headers of each section, the words in bold in the text. Now, the other thing I need to tell you is I'm going to use the in context definitions of those words on the lecture quiz. And what that means is I'm going to use the definition from the words around it in the textbook. So don't Google the words and write out these long definitions that aren't the words from your book, because I'm not going to ask you those. I'm going to ask you the context words, definitions from around the context words in the chapter itself. So that's first for chapter 13. Yes, there are also concepts that we need to cover for chapter 13. I will post a module. I was just looking in my course from the spring, and there's a whole bunch of things that I can bring over that are note outlines and videos from chapters 13, 14, and 15. So I will do that in the coming days. Right now, the other thing that I would like to talk to you about is the lab. And as I mentioned in the announcement, there's going to be 2 lab modules. The first module is going to be safety and care and use of the microscope. Safety, because you have to be able to follow all the safety procedures. There's also a little video on how to make a 10% bleach solution for disinfecting purposes. And then there is a lab safety booklet for you to review and answer some multiple choice questions. It's a quiz, lab safety quiz. Module 2, for the lab module 2, will not open until after you've completed the lab safety quiz and after you've uploaded your picture results and answers to the questions for the brine shrimp lab. So come with your microscope kit. The introductory lab that comes with it is growing some brine shrimp and then looking at them under the microscope. And I would like you to complete that lab, basically to learn how to use your little My First Lab microscope. Also, to help you prepare, it's practice of making some slides. Please use the plastic slides that are in the little slide box, not the glass slides that came separately. The other really important thing for the lab modules is once you complete lab module 1 and you can move into lab module 2, I'm going to ask you to complete, first thing you're going to have to do is grow 4 different bacteria. And that they're called the micro quick kits. And that is in your packet, in your lab materials that you received in your box. You're going to need to grow those 4 organisms before you can complete the rest of the labs. Now, the tricky part is you need all 4 of those organisms to complete the rest of the labs. So you have to keep them alive for as long as it takes you to get through the rest of the labs. So the rest of the labs include doing a simple stain, a gram stain, so that would take you maybe an hour, setting up streaking by isolation, and setting up transfer. Actually, I do the transfer first and then streak for isolation. And then there's a biochemical testing lab to do. So you're going to need to set up 5 labs: the streaking, the broth diluting or moving transfer, the transfer lab, so transferring the bacteria into broth cultures and slants, I believe they are, and onto Petri dishes, and then streaking for isolation on Petri dishes. And the gram stain and the simple stain, you need those bacteria. So some students in another section, not in our section, but in another section who started the lab, accidentally killed their bacteria and threw them out after they completed the first lab. And then they didn't have any cultures left to complete all of the labs, the rest of the labs. So this is really, really, really important that you're going to need to first set up those cultures, the microquick cultures, and each of them has a video embedded in it from Carolina themselves that shows you how to do it step by step. You're going to set up those cultures, and then you're going to use the cultures to do a transfer lab, to do a streak plate lab, and then those streak plates and the transfer broth will give you material to perform the simple and the gram stain, because you'll be able to use the bacteria that you've grown. And also then you'll be able to use the bacteria you've grown for the biochemical lab. So it'll go fairly quickly, but you should probably try to plan once you hit the, once you complete lab unit, the first lab module, and you're getting ready to do the second lab module, I would strongly advise that you read all of the instructions and you get yourself organized before you start, because you do need to keep those cultures alive to do all those labs. Now, setting up the transfer lab and the streak plate lab are relatively simple, and you set them up and then you let them grow. So you just set them up and then they're done until you need to look at the results, which is usually 24 hours and then probably 48 hours. They probably ask you for results at both time points, at 24 hours and 48 hours. So you do kind of have to plan, okay, for the next 4 days, I'm going to do one lab every day, or I'm going to set up these labs, and then the ones that I have to read the results, I have to make time to read those in the next few days. So I wanted to explain that to you and warn you about that, because I don't want you to throw away your cultures, because once you throw them away, or if you kill them with bleach, then you won't have any cultures left for the rest of the labs. So that's really important. And I'll post that again when I'm at the top of the lab module too, but it is going to require you personally, each one of you, taking the responsibility of organizing yourselves, because I can't say, oh, everybody has to do this lab, this lab, this lab on these days, right? I'm not in control of your individual personal schedules. And so that's why the labs are more flexible, and they're all going to be due the same day, so that if someone can do them this week, great. If you can't do them until next week or 2 weeks from now, fine. But you're going to first complete the lab safety and the microscope module, which I have to put the quiz in for lab safety, and then I'll open it up. And then the second set, you'll have to complete kind of all together. So I wanted to let you know that. That's really important, and it's new and something that you haven't done before, I would guess. So I wanted to let you know that. And then I also wanted to let you know that I will post, I think I have note outlines for, I'm not sure about 13, but I know for 14 and 15, and I know there's videos from the spring for 14 and 15, not sure about 13. If there isn't one for 13, then I'll be doing a lecture for 13 on Monday. I want to check that first, and setting up the labs is a higher priority for us right now than continuing on with the lecture material today. So I was kind of planning on today being a short Zoom meeting so that I could work on getting those labs open and available for you, because some of you, I think the brine shrimp you have to grow for a day or two as well before you can look at them. So you have to rehydrate them, keep them a little bit warm, I think, and then let them grow for a day or two, and then do the microscope lab. So that will take you a few days to complete. And the lab safety will just be read it, learn how to make the bleach solution for disinfection, and then take the quiz. And then again, after you've completed the lab quiz, the lab safety quiz, and the microscope lab, which will be the brine shrimp will be a separate little lab report, then the prerequisite for you to getting into the Carolina labs is going to be having completed those first 2 labs. So you're going to have to do those first, and you have to learn how to use the microscope, because you're going to do a gram stain and a simple stain, and you have to be a little bit adept at using the microscope before you just try to look at bacteria. Okay, questions that anyone has about what I've covered, or if you have questions for the quiz, that's fine too, because I know a lot of you joined after we started talking. Yes, no, no. Nothing. Okay, then if no one has any questions, I'm going to end the recording.